Okay, hi everyone. Welcome to the 8 Notes Biology September revision lecture. I hope you guys are all doing really well. I hope you are having a lovely sorry, I've just got a bit of a cough. But I hope you're having a lovely holiday so far, or as lovely as it can be with all the HSC prep happening. So I hope everything's going really, really well. Welcome to the lecture. It's so good to have you guys here. So a very warm welcome from all of us here at 8 Notes. So before we begin, just quickly introduce you to some of the amazing resources that we have to offer here. And that includes study notes. So if you're now, looking for some last minute study notes to add to your collection, our website's a great place to check it out. So we've got free study notes that students post their own study notes, and it's a great way to consolidate your notes if you're looking for that. We also have our lectures happening this week. So with the lectures, we've got our revision lectures like the one happening today, and also Head Start lectures. And Head Start lectures are for next year's class. Well, the current year 11's going into year 12, and it kind of covers the sort of the first like the content for term one. So if you kind of want to brush up on that, feel free to attend those lectures as well. So we also have our lectures, our discussion forums online. It's an amazing community of like-minded students. We have our newsletters, our ADA calculator, and also lots of articles that range from study tips and strategies to like uni tips and just like sort of general student stuff that I'm sure a lot of you will find really helpful, that I personally found really helpful because I love the website, love using these resources. So definitely check them out on our website. So let's get started today's content. So firstly, before we do that, I'll introduce myself. So my name is Aditi. I graduated in 2021 as a Dux with an ADA of 95. So the subjects that I teach include English Extension, Advanced, PDH, PE, and Biology. And so I'm currently at the University of Sydney studying a Bachelor of Science and Bachelor of Advanced Studies in Medical Science as a Danielle Scholar. So I'm majoring in medical science and political economy. Now, if you have any questions about uni, about any coursework or anything, please feel free to ask those as well. I'll try my best to help, to help you out. I mean, I'm still trying to figure out all of that stuff myself. But if you have any questions, I can always try my best to help with that. But yeah, and I love in general reading good shows, sunny days, and currently it is shaping to be a very sunny and a very hot day for me. It is like shining bright. It's amazing. I'm loving the summers or the beginning of the spring. Yeah. So it's been really exciting. So let's get started. We have lots to get through today, and I know you guys will probably be really excited in a way. I guess maybe not quite the best word, maybe not jittery or nervous. I'm probably a bit nervous with the HSC exams coming up, but look, my aim today is to essentially revise the main components of the HSC biology syllabus, right? To get you ready and confident for sort of the main like across the main talking points of the syllabus. Now, please make sure that you check anything that does not make sense. Ask me. I am here for questions. I can definitely answer your bio questions. If not, any uni technicality questions, definitely answer your bio questions. I've been doing this for a while. I'm studying biology at university now, so I think I'll be okay with that. And I'll be answering your questions live. So this is obviously recording, and I'll be live answering your questions. And that reminds me that you'll be able to firstly view this recording afterwards as well. So come back to the page and view this recording afterwards. You will also be able to access the slides that I'm going through, like just underneath your screen, or well, underneath my sort of screen where this video is playing, just underneath, probably on your left hand side corner, you should see the slides, a button to like click and download the slides. And what else? I'm just remembering I'm forgetting anything. Uh, yeah, and I'll be, like I said, I'll be live to answer any questions. So please don't be yeah, please don't be shy, please don't hesitate to ask any questions. No question's a silly question. Even if you feel like, you know, you should know the answer to this but you're not too sure, ask. Always ask. Finally, remember that, you know, you're gonna smash this out. You have to give yourself a bit more credit. You guys have gone through an amazing year. Or what I hope has been an amazing year. And you know, biology is definitely a very content heavy subject. So if you have stuck with it until the end, cheers to you. And we're gonna go through this together. So hopefully today you'll walk away feeling um, a bit more confident and a bit more ready. And I'm sure you guys are obviously, you know, looking forward to, maybe not forward to the exams, but certainly the period after that, the summer, the schoolies period after that. So I'm excited for that for you guys because I think it'd be really fun and the weather's shaping up to be amazing. So yeah, let's get started, shall we? So the HSC syllabus. Now, hopefully we're all very familiar with this. If you've forgotten something, that is okay, it happens. But now's a good time to like really solidify this and glue it in your minds. So we've got module 5, which is heredity, and it's in module 5 we're focusing on reproduction across across different types of organisms, and we're focusing on also the different types of reproduction, different and like we want to know examples for it. I just might just grab a tissue, sorry. Alright, let's see if I can find a tissue now. I have to open a new box. Please excuse me, sorry, I'm just recovering from a flu, from the viral, so yeah, please excuse me. Alright, so where was I? Okay, so back to module 5. So module 5, heredity, we are looking firstly at reproduction. We're looking, and the key question is, how does reproduction ensure the continuation of a species, right? So you have to look at different types of reproduction, you have to look at different types of fertilizations, and we want to know examples across different organisms as well. Cell replication is now looking at those molecular processes of mitosis and meiosis, and how is our DNA being ready? You know, it's being ready, being made ready rather, before we can go ahead and have DNA and protein synthesis. So remember, replication happens before that, and then we look at genetic variation, and finally we finish off by looking at inheritance patterns in a population. Module 6 is obviously the smallest one out of the 4, and it's also the one that has a lot of similarities, I would say, across like it has a lot of overlap between the other 3 modules. So mutations, we start off by looking at the different types of mutations and their causes and their effects as well, and that links really closely back to module 5. Then we look at biotech. Now biotechnology also kind of links back to module 7 and 8 a lot, because we look at technologies and how they can be used to treat different disorders. And then we also look at genetic technologies. So that's module 6, and like I said, there's a lot of overlap between the other sort of modules with that one. Now what you'll find is that you get a lot of intermodule, you get a lot of you get a lot of intermodule questions as well, especially the higher mark questions in biology. They do tend to ask you to apply your knowledge across like 2 different modules, and those questions are very popular for those higher marks, because it sort of tests your knowledge if you can apply the content that you've learned from these 2 different topics and synthesize it and bring it together. Module 7 is infectious diseases, and within that, we're looking at what are the causes of infectious diseases. We're looking at responses to pathogens. Then we look at immunity, so we get like a very surface level dive into immunity, which is always very exciting. If you're into sort of the, and not quite the anatomy, but definitely like the physiology and sort of the anatomy side of things. And also we look at prevention, treatment, and control of infectious diseases, and that one has definitely been more relevant in contemporary times because of the fact that we have gone through a pandemic just before, just a couple of, well, it's funny now because we're still kind of, it's still there but it's not kind of there, but we've gone through a pandemic, so we know like a lot of it is pretty intuitive when it comes to like treatment and control and like prevention, which like mainly focuses on like um, hygiene and quarantine and so on. And like I said, we're all pretty familiar with that. Finally, module 8 is non-infectious diseases. So this is where we now look at we start off by looking at homeostasis, which is a very interesting topic, and that's basically your body's machinery of your body's sort of internal machinery and its purpose to sort of maintain a stable internal environment despite the changing external conditions. We also look at the causes and effects of non-infectious diseases. We look at epidemiology, and then we look at prevention, and finally technologies and disorders. And then in technologies and disorders, again, you get a bit of a taste of, again, like a very surface level taste of anatomy and physiology, in the sense that we sort of see the structure of your, so the three that you have to do according to the syllabus are your ears, eyes, and kidneys. So it's like you get to sort of see how does the ear actually work, how is it structured, and then what is the function of it. So it's quite interesting, all of that. So yeah, that's the that's the whole biology syllabus. But there is, like I said, lots of intricacies within each module, so let's attack that. um so let's have a look. We're going to look at the key concepts today of reproduction. We're going to look at DNA replication, we'll look at protein synthesis, and then we'll look at inheritance, which is Punnett squares and pedigrees. So we won't quite be covering meiosis and mitosis today. So yeah, we won't cover that, and we won't cover DNA sequencing and profiling. So you can obviously access, you can obviously access past lectures as well, where we've gone through this in a lot of detail. We also have there's also lots of good online videos, like for example with mitosis and meiosis. You'll find lots of really nice animations on YouTube if you really want to sort of, if you're finding it a bit hard to understand what's happening, just because you're reading over the steps but it still doesn't quite make sense. I would 100% recommend watching a YouTube video on it. And one that I really like is like the channel that really worked for me, that I really liked for all of these bio concepts, was the Amoeba Sisters. They're really good, and like and the animations are really nice, and so it makes a lot of sense because it's like, it's very visual. Yeah. Alternatively, Crash Course is also a great website to use. They can sometimes go in a bit of detail, but it's still really nice in terms of the explanation. So definitely check those out. But let's get started with reproduction. So give me one sec. Okay, so we've got essentially pregnancy hormones. We're going to look at that part of the dot point because it's generally the one that's not overlooked, but I think it's one that people tend to forget the most. So we've sort of categorized the main hormones that are involved in birth into 4 categories. The first one are the bleeders. These rule the menstrual cycle. So you've got FSH, which is the follicle stimulating hormone, and LH, which is the luteinizing hormone. So they regulate the menstrual cycle. And then we've got the controller, which releases the hormones. And so we've got HCG, which is the human chronic gonadotrophin, and that is, you can also remember it as a hormone control guy. But yeah, that's a controller. So the human chronic gonadotrophin releases these hormones, and then once the mother is once the egg's been fertilized, this is where we've got the growers now. They stimulate growth in the mother and the baby, and that's estrogen and progesterone. And finally, when it's time for labor, you've got the pushes, you've got oxytocin, you've got relaxin, and you've got progesterone. So these are all really important, and you want to know them. These are just basic summaries for you guys to remember what the sort of the links are between each, but I would recommend to go ahead and jot down actual notes of, for example, what is estrogen doing when it is stimulating that growth in the mother and the baby. What is HSC doing? What is oxytocin and relaxin doing, right? So I would definitely recommend to go ahead and jot down more detailed notes. Just a couple of points, but I just want you to remember also the categories here so that you can actually remember them. So let's move on now to DNA replication. DNA replication is obviously, it's a long process in the sense that there's a lot going on. So we need to remember some of the really key important terminologies and, you know, enzymes and stuff that are involved. So we start off, the first process is initiation. So in initiation, the DNA is unzipped by helicase, by the enzyme helicase. So that's where, that's helicase right there. Oh, I'm going to get my little pointer pen thingy out. There we go. Alrighty, so that's helicase there. So helicase comes ahead, oops, and unzips the DNA. And then what happens is with the second process is elongation, so it moves forward, which means that as it's unzipping the DNA, we've got DNA polymerase, which is going to bind at primer sites. So this is DNA polymerase, it's going to bind at primer sites. Now, primers are our RNA primers that are going to come and show the polymerase where to bind. So that's what they are. So they are RNA primers. It is, you know, DNA. Oh, why is this not letting me make, like, right? Don't have the best handwriting with the cursor, but let's try. Here we go. So even though this is DNA replication, what is happening is the primer that is coming here and showing DNA polymerase where to go is an RNA primer. So RNA. Alright, so RNA primer. Cool. So what happens is once it binds, it then starts to read the strand and attach the free-floating nucleotides. Now, the really important thing here is that we need to remember that DNA replication is happening in the 5' to 3' direction, right? So it's happening from 5' to 3'. So one strand is fine because it's going forward this way, but one strand is coming backwards. It's coming backwards. So DNA polymerase moves along the old strand in the 3 to 5 direction and creates a new strand in the 5' to 3' direction. So as you can see here, it is coming. I'm just trying to, where is it? Let me just rub this bit out for a second, sorry, so I can. Okay, here we go. And let's go this way. Okay. So what happens is the 5' to 3' direction is fine because it's, you know, that's the way the DNA polymerase goes. But when it's coming back in the from the 3' to the 5' direction, what happens is is that it is going to be putting on the nucleotides backwards. And because of the fact that it's putting it on backwards, we get something called Okazaki fragments. And that's just that you've got these groups of basically these groups of nucleotides. So there's like gaps in between. And so after the process is finished, we have we see after like polymerase finishes the whole process, it reaches the end of the molecule and it falls off. And then what happens is the Okazaki fragments, they are filled, and so it's like a proper strand of DNA with like no gaps in between. But because initially the DNA strand is going backwards when the nucleotides are being added, there are gaps in between which are then filled by the enzymes. So that's elongation. And then termination is when polymerase reaches the end of the molecule, it falls off, and that's it. And so then the strand then the strand essentially will recoil into a double helix. So remember, it's organized into a double helix because of the fact that there's so much information that we have to preserve. And so as a result of that, once it's all over, it's going to twist back into the double DNA helix, and proofreading also will occur by nuclease enzymes. Proofreading is really important because of the fact that there can be obviously mistakes, and that's where proofreading comes in. This process is something that we need to know really well, because we'll come back to this when we look at mutations and how mutations can affect the DNA and the genome. So that's DNA replication. If you have any questions, please ask. There are no silly questions, please ask. Alright, so okay, alrighty, so let's have a look now at polypeptide synthesis. So polypeptide synthesis is basically, think of it as a recipe to create proteins, and a kitchen is going to be the ribosomes, right? So in the cell, it's going to be the ribosomes. That's where we go to create our proteins. We prep our stuff. Let's say you prep your stuff in the living room because you're sitting and you're just like chopping up the veggies while you're sitting down. But then when you actually have to cook, you get up and you go to the ribosomes. You go to the kitchen to cook. Okay, think of it that way. So that's what happens. The first process to transcription happens in the nucleus. Second process, translation, happens in the ribosome. So what is actually happening? So what happens is then essentially step one of transcription is that we start off with our DNA strand, right? With a double DNA strand. So RNA polymerase now, because now we want to, what we're doing now is we are going from DNA. We're going from oh, this is not letting me. Looks like my computer's had enough of my handwriting, because I've been doing it for my other lectures as well, and it's not the best. But let's try this. Okay, we're going from DNA to RNA. So now we've got RNA polymerase coming along. And what RNA polymerase is going to do is it's going to come along, and we're going to open up the DNA double strand. Now, once we open it up, we have messenger RNA come through. Now, the main difference between RNA and DNA is that firstly, RNA is just made of ribose sugar. DNA is deoxyribose. That's the big difference between the two. So the sugar in the backbone is different for RNA and DNA. And then on top of that, in RNA, you've got uracil instead of thymine. And the reason that happens is because of you don't need to know this, but if you are interested to know why it changes from thymine to uracil in RNA, is because of like mutations happening to thymine, and as a result it becomes uracil. No, mutations happening to it affects cytosine and thymine, and then as a result it becomes uracil. So you don't have to know it in a lot of detail. It's part of something what it's my learning from uni this year, second year, so you definitely don't have to know it, but it's just there in case you want to know it. It's because of mutations essentially that we see we see that distinction between the two. So, but yeah, but you need to remember that it's actually uracil in RNA, so it's going to be uracil paired against adenine and not thymine. So let's have a look now. Essentially, here we are. So mRNA comes in, and what mRNA is going to do is again go from 5' to 3' direction, and it's going to start making a template strand. But now every time, instead of a T, there's going to be a U. Alrighty, so instead of a T, there's a U. So we have that replacement here. One is the RNA polymerase. And it's RNA polymerase that is essentially taking the whole process through. That's really important. RNA polymerase is taking the whole process through. And so we've got a coding strand, a template strand, and then the RNA there. So let's have a look. Alrighty, now wood transcription. So once the mRNA is gone and we've got that mRNA strand with uracil on it, so sorry. What happens next is that a process called splicing occurs. So splicing is oh goodness. Give me one sec. Okay, now we'll cease this. So splicing is basically when we get that mRNA strand, it's got everything in it. So what we need to remember is that a whole DNA is not coding for protein. So or not. I swear I was fine yesterday. I don't know what happened. But anyways, let's focus. So the thing here is that a whole DNA does not code for protein. There is junk DNA, there is regulatory DNA, and then there's coding DNA as well. But coding DNA is actually not a lot, so it's quite interesting. So what happens is that when we get that mRNA template, right, it's got everything in it, but we don't want that because we need the code to then make the protein, right? Because so let's say I've chopped up everything. I've chopped up all the vegetables, but I want to make pasta, so I'm not going to put potatoes in pasta, and I'm not going to put, I don't know, beans in my pasta, so I'm going to leave the beans and the potatoes out. I'm only going to take the capsicum and the onions with me into the kitchen. So that's kind of what's happening here. So what happens is we take out, so we've got the exons and then we've got the introns. Alrighty, so it's the exons that we want. So we take the exons, as you can see here, and we leave the introns out of it. Alrighty, so we leave the introns out, we cut the introns out, and we take the exons. We take our coding DNA. And once we do that, we end up with mature mRNA. And we're going to use that mature mRNA to make our proteins. But we've still got one more step to go before we could do that. So, now, that's transcription. Transcription is going from DNA to mRNA. Translation is now when we go from mRNA to the proteins, but this is where tRNA comes in because it's translation. So, let's look at this image right here. So we've got a T, so it's a tRNA, and it's got 3 anticodons. And each the anticodon, so it's got anticodon, which is made up of 3 RNA molecules, or 3 RNA nucleotides basically, and it codes for an amino acid. Now, hopefully you would have all seen that table that sort of shows the different combinations of the amino acids, right? Like what codons make what amino acids. So what happens now is that within the ribosome we've got all of our different amino acids floating around, let's just say, right here. We've got them here, and they're coded for, goodness, why is it doing that today? And they're coded for by different, by a group of, or by like a specific anticodon. Let's say that it's coded by a specific anticodon. So what happens is, now I've come to the ribosomes, I've come to the kitchen with my capsicums and onions, right? So that's all I need to make my pasta. So I've come in, and now what we're gonna see is that the as the mRNA comes into the ribosomes, the complementary anticodons attached to the codons. So the ones on the mRNA are codons, and then we see anticodons attached to it. So if we've got ACG attaching here, that means this one is going to be, what's this one going to be? If you said uracil, you're correct. If you said thymine, you're kind of correct. But remember, it's RNA, so it's going to be uracil. So it's going to be uracil. C's going to bond to G, guanine, and then G is going to bond to C. So as a result, this thing here, so we've got our codons. They are basically going to bind to their specific anticodons. It's the same, it's the same sort of nucleotide bonding here. So this is what's happening. As a strand is going along, the right anticodons are going to come and bind. And now and this is what's happening when they come and bind. So remember, we've got amino acid here, and now tRNA is bringing those codons, those anticodons, to the mRNA. So remember, this is it is, seriously, why is it doing that? Here we go. I'm going to try and write. So this is mRNA, and this is tRNA. Alrighty. Remember, these are codons. It's annoying me now. Why is it making their straight lines? And this is anticodons. Is it gonna make it this time? Let's see. Oh god. Made it twice this time. Wow. Okay. These are anticodons. Alrighty. So that's that. Alrighty. So we've got our tRNA. We've got our anticodons, and then we've got mRNA, and we've got our codons. Okay, perfect. So finally, what happens is, once they match up, and once the codons and the anticodons match, we get our amino acids, and these are going to be our amino acids. And then our amino acids are going to form polypeptide chains, which are going to form proteins. And remember, there are different structures of proteins. There's primary, secondary, tertiary, and quaternary structures, and proteins are obviously really important because they have a huge role to play in structure and in function within our body. Our enzymes are proteins. Your hair, your nails, everything needs protein. You need protein obviously as a diet thing as well, so it's part of that as well. So it's really important that you understand the whole process. This was obviously like a little crash course with to sort of explain the basics of the process in case you may have forgotten, or just to re-jog your memory. But remember, that is the key there. But I would obviously recommend to go back and look at your notes, and if you don't have them, add to them and make sure that you understand the process. And I would also recommend watching some YouTube clips because they are really good in terms of their animations. And the animations are really, really helpful in having like a really good visual picture of what is happening. As you could see, I mean, I could try to animate, but you saw how my handwriting turned out, so I'm not sure how much hope we have for animating, guys. But I hope the explanation helped. All right, and check out those videos to get a better understanding of how it all looks when it's happening. Alrighty. Yeah, so remember, you've got, like, it's not a process that's happening all in the same place. It's a very dynamic process. It's a very dynamic process remember we start off in the living room when with our DNA, we're making an mRNA copy. I'm chopping down all the vegetables that I've got, but then I realize that hey, I'm making pasta, so I only want to put capsicums and onions in my pasta. So I'm only gonna take that with me, which is my mature DNA, which is my mature RNA, only the bit that's actually going to code for the proteins that I want. And so I take that into the kitchen with me and use that to cook. And that's what's happening there. So let us move on then. Okay. And so, yep, that results in our protein. And we've kind of already spoken about the sort of the key processes. Now remember, after translation we do have post-translational sort of checks and edits that are happening so that we can get the mature protein ready. So let's have a look at, so this is basically a little summary for you of all the processes. So what's happening in terms of where we sort of start off, what we end off with, and then how we begin again with translation. So just quickly go through it again. In transcription, remember we're in the nucleus, RNA polymerase binds to the promoter sequence, and then from there it's moving along the DNA strand. Now it's only going to unbind a small region of the DNA. So the reason for that is that we are not, remember the whole DNA is not coding for proteins, and then on top of that, specific regions code for specific things, and we only ever need to code for specific parts or specific proteins. You don't use the whole thing. So it's going to unbind a small region that we need. The RNA polymerase will read this strand and it will make the complementary free floating nucleotides to create an mRNA chain. It's mRNA because remember RNA we're using uracil. And then once it's done, there's a terminator sequence that ends the transcription of the DNA, and it releases the mRNA molecule. And then remember we have a splicing and post transcriptional, that should say transcriptional, not translational, modifications are made to that mRNA because we don't want the whole thing, we just want the bit that's coding for the DNA. And so we only take that with us to the kitchen, which is our ribosome. So that's where translation happens. So translation, think of transcription as like copying the recipe, and then translation actually translating the recipe into the food. So translation, mRNA docks to a ribosome. The ribosome matches that complementary tRNA molecule to the mRNA, so it matches the codon to the anticodon. Alrighty, and so after that, when the molecules dock, right, when the RNA molecules dock, a polypeptide is formed. So those amino acids, they start to form these polypeptide bonds, and that's like actual chemical bonds so that they stick together. And once they have done that, there's a polypeptide chain of amino acids. And when a stop sequence is reached, that's when the ribosomes release the mRNA. So they let that go, and they also let go of the polypeptide molecule. And then the polypeptide folds, right? And then it undergoes post-translational modifications and result in a mature protein, which can then be used in the cells for a diverse range of reasons. Okay, let us move on then. Okay, so this last bit of module 5, inheritance. This bit, I would say the best way to understand this bit and the best way to actually answer these questions is to just draw diagrams. That's it. Draw diagrams that are going to be really, really, really helpful. Yeah, and just do practice questions, because I mean there are definitely with this, and we'll go through, there are definitely like tips for it, and you can see like different patterns and sort of generic tips, but the best way to feel confident with these is to actually do lots of questions and draw them out. Whenever you get a question, draw these things out. Don't ever try to like do it in your head, always draw it out. So remember, there are two things that we do here. The first is we draw pedigrees, and the function of a pedigree is to actually trace back different characteristics and traits within generations, and it allows us to see like the overall pattern of, you know, how is the disease sort of or how is the trait rather being passed on, who's getting it, how, you know, is it like how often is it showing within the lineage, and then Punnett squares on the other hand are actually to rather see how genes can pass from parent to offspring. So pedigrees are helping us trace phenotypes, things that we can actually see, or conditions that can be seen, or not seen and like, you know, diagnosed and stuff. So those are actual like the phenotypes. Punnett squares are genotypes. You know, we are sort of looking at potential combinations of what alleles can, you know, an individual get from like the different combinations of their parents. So this is giving us information about potential genotypes and phenotypes. So whenever you get a question like this, the checklist is you want to have a key, you want to have parental phenotypes and genotypes, and then you want to fill in the Punnett square, and then you want to comment on the offspring's genotype and their phenotype, and you want to do that both. And I mean, questions can like suggest that if they want you to do it as a percentage or as a ratio. If they don't suggest it, just do it as both, because again, it's not hard to like just convert between the two. So just do it as both to be on the safer side. So let's have a look at Pedigree's checklist. How do we actually determine if a disease is autosomal dominant, recessive, X-linked, dominant recessive, or Y-linked, or mitochondrial? This is a really interesting one, and best way to sort of get better at this is to actually go ahead and do these questions. You'll find lots of them, and you'll find actually questions not only in the multiple choice but also like short answer questions in the past HSCs where they've actually asked you to draw these out and to be able to comment on, you know, why would a certain condition be X-linked versus autosomal. So let's have a look at what the traits are of each. So if now remember, autosomal means that it is not on the sex chromosome. X-linked is that it is on the so autosomal means that it's not on the sex chromosomes, it's on the other 22. X-linked is that it's on the X chromosome specifically, and then Y-linked is that it is on the Y chromosome. So autosomal recessive, so if a condition is autosomal recessive, it will appear in both sexes with equal frequency, and it will affect, and affected offsprings are sometimes born to unaffected parents because it is recessive. That's why we can see that unaffected individuals can have affected offsprings. It's because it's recessive, so it can like skip a generation. Autosomal dominant, again, because it's autosomal, it's gonna appear with equal frequency in both sexes. However, affected offspring will have at least one affected parent because it's dominant, because it's dominant. It's like, for example, it's you know, like homozygous, so it's gonna be there. At least one parent will be affected. So if something's dominant, let's say the parent is heterozygous, they're still going, and it's a dominant condition, they're still going to be affected because it's on that dominant chromosome, if that makes sense. Because it's hard to explain that without like diagrams and actually like drawing it out. But again, I'm not quite gonna draw it, but you know what I mean. You know, for example, if you go H, right? If it's dominant, that's heterozygous. But if it's a dominant condition, this person with this genotype is still going to be affected. Then X-linked dominant. Now, X-linked dominant means that it's on the X chromosome, and now because it's on the X chromosome, we can generally see that females will be more often affected. And because it is dominant, it's not going to skip a generation. You're going to see it in every generation. And then affected sons must have an affected mother. So if it's X-linked dominant, one of the one of the main ways to tell if it is X-linked dominant is if it is going from an affected mother to an affected son. Um, and if it is an unaffected father, for example, if a father is affected, he will pass it on, the trait, onto all their daughters. So the way this works basically is that, remember, because it's X-linked dominant, firstly, dominant, when it's dominant, you know that it's not going to skip a generation. Recessive, it's going to skip a generation. However, in terms of X-linked, a good way to tell it is that, firstly, females are more affected. Secondly, affected sons must have an affected mother. And the reason for that is because sons get their X chromosome from their mothers, because they only have one X chromosome. Whereas so they only get one X chromosome, whereas daughters will get, like, they'll have so women have two X chromosomes. So what that means is that an affected father, because the father will only have one X chromosome, so he's only going to pass on that one X chromosome, so father will pass the trait onto all of their daughters. That's the logic behind it. Then X-linked recessive. Now, with X-linked recessive, more males than females are affected. That's another key way to tell. And with X-linked recessive, approximately half of the carrier mother's sons are affected. That's another, like, really obvious way to tell if it's X-linked recessive. So I would definitely, like, jot these down, because it's like these patterns are the best way to tell. Otherwise, there are so many possibilities, and it's hard to sort of figure everything out within the limited minutes you have to attempt the question and the limited information you're given. So I would use these little prompts as a way, in patterns, to seek out and to eliminate other options. So, and remember, X-linked recessive, it can skip a generation. Y-linked dominant, we don't actually see a lot of questions about that, but you can obviously see them, and that's Y-linked. Remember, only males have a Y chromosome, so only males will be affected. It will be passed from father to all sons, because they get, like, all, what do you call it? They get the Y chromosome from the father, and it does not skip a generation because it's dominant. Mitochondrial is a trait which is inherited from the mother only, and all children of a mother are at risk to be affected or to be carriers if it's mitochondrial. Again, you don't see it as often. These are sort of the main ones, these four, but it's good to know these two down the bottom. Let's move on then to module 6. Look at us go. Okay, module 6, we're going to go quickly through everything, so let's have a look. Mutations. There are essentially two types. Firstly, there are structural chromosome mutations. These are mutations to the chromosomes themselves that have happened during, for example, during meiosis, right? So these are actual chromosomal mutations. So there are 4 types. There is deletion, there is inversion, there is translocation, duplication. And as the name suggests, in deletion, a chunk of the chromosomes cut down on. Inversion, it's been inverted. Translocation, like it's been switched. And then duplication is that, it's been added on again at the top. So when there is a mutation in the chromosome numbers, where's my thingy? Okay, here we go. When there is a mutation in chromosome numbers, essentially we classify that as either aneuploidy or polyploidy. So aneuploidy is when the overall number of the chromosomes is different between the parent and the offspring. So the offspring has one extra or one less chromosome. And Down syndrome, for example, is an example of when the offspring has 47 chromosomes instead of 46 chromosomes. Polyploidy is when an organism contains more than two sets of homologous chromosomes. It's not the overall number. I mean, obviously it will affect the overall number, but it's actually like this, for example, 3 sets of chromosomes. So triploidy is a condition in which fetus has 3 copies of every homologous chromosome instead of only 2 copies. So that's chromosomal mutations. Now, point mutations. Point mutations, essentially, these are the ones that are happening within the sequences of the codons. So let's say we start off with a normal codon sequence of HEC, AA, G, double G, CU. So that gives us these 3 amino acids, all right? Now, if there's substitution, let's say during the, and now remember, mutations can be chemical, they can be electromagnetic, right? So what happens is with mutations, and mutations can cause the like nucleotide sequences to shift within your DNA, to change. And so let's say that a mutation has happened, and it's happened and it's not been detected by like the error system that actually checks everything and makes sure everything is right. So it's not been detected, so there's a substitution that has occurred, and instead of the AGC, the G has been replaced with a U, and that substitution has actually caused for the amino acid here to change. That's substitution mutations. Frame shift mutations are when there's been a shift of the actual strand, which has caused like a downstream effect. So there can be insertion, where another nucleotide has been inserted within, and that can again lead to actually, and you can see here, that can actually lead to the whole sequence, the whole 3 amino acids changing. And in deletion, one of the parts has been deleted, and that causes also obviously 1 amino acid to be removed and the other to be changed completely. So then how do we actually classify these mutations? So we've got 2 types of mutations, 2 types of point mutations. We've got substitution and frame shift. Frame shift can include insertion and deletion. Now the effects, the way we classify them, are either missense, nonsense, or silent. Now, if a mutation is missense, that means that mutations which edit the codon sequences, so there has been a change in the amino acid that has been sort of, that's been a result of the whole process. So there's been a change in that amino acid. So now instead of getting SER, we got ILE, right? So that's that. Nonsense is when we introduce a premature stop codon. And that is seen here with the UAG. That is a stop codon, and so we've prematurely stopped the process. Finally, a silent mutation is when there has been a mutation, but it's actually not affected the amino acid. Because remember, there can be more than, now think back to the table that you would have looked at, when you're looking at like converting between the anticodons and the amino acids. So think of the fact that there are more than like one there's more than one anticodon for some of the amino acids. So here we have AGU will also result in cell, whereas AGC will also result in the same amino acid. So that's what we have, that is what has happened, but luckily there has been no effect on the amino acid. So you want to know the 2 types of mutations, structural versus point mutations, and then within each you want to know the categories as well and the effects. Okay, so that's biotech. Let's have a look now at recombinant DNA technologies. So, sorry, did I say that was biotech? No, that's mutations. Whoops. So go straight to genetic technologies, and we'll quickly talk about some of the key key technologies here. So we've got recombinant DNA technology, and recombinant DNA technology is when we're joining DNA from two different species in order to produce a new genetic combination. So it's basically a simple version of this is, for example, we take a recombinant plasmid. So a plasmid that contains the genetic information of two or more organisms, right? So we've got the first plasmid, and what we do is we open up the plasmid, we open it up, and we digest it with restriction enzymes. So what restriction enzymes do is that restriction enzymes will open up the plasmids at like a specific point that we want them to open up on, and so it will open it up and we'll insert a target gene there. So once we insert it there, we then join it up with DNA ligase, which is kind of like a glue basically. It's obviously an enzyme, but it works like a glue to stick it all together, and then we get recombinant plasmids. So a recombinant plasmid will have the DNA, will have the sort of obviously the genetic qualities of the plasmid itself, and then also we have the target gene which is inserted in it. So yeah, so they're like, for example, with restriction enzymes, a quick note on that, there are different types of restriction enzymes for different types of plasmids, and generally for different types of plasmids, and they cut the plasmids at different points, at different base pairs. So it's wherever we want to insert a target gene, we use a specific restriction enzyme to cut open the plasmid at that specific place, so that we can get a recombinant plasmid that actually works and sticks together. Alrighty, looking at the production of transgenic organisms. So transgenic organisms, essentially, so we start off with an individual. We are going to isolate DNA from that organism. We're then going to sequence that DNA to sort of see, to get the whole genome. And then we're going to identify that gene sequence. We will then create lots of copies of that gene sequence using polymerase chain reaction. So it's going to create lots and lots of copies, depending on how many. Generally, it's like you run 30 rounds of it, so it creates lots and lots of copies. And once it does that, we then verify the gene. So we make sure that we've got the right gene. So we use gel electrophoresis. Basically, what happens is we put in, these are the bands, the base pairs that we put in as like a benchmark, and then we test against that 1, 2, 3, 4, right? So we test which one matches up the most with this. So which one matches up the most with the benchmark here. So that's that, gel electrophoresis. And then we put the gene into an organism. And we can do that by doing the whole restriction enzyme cloning. So by making a recombinant DNA. And then through that, we then transform the organism. So we can also put the gene into, so we could do it using restriction enzyme cloning. We can also do transfection, so we can directly insert it into the nucleus, or we can use a microinjection as well to insert it in. So there are 3 ways in which it can be done. So if you have any questions, please ask. But I'm hoping this process made sense. And like I said, if you have any questions, anything I can help out with, please ask. Okay, so a huge part of the syllabus is now to also look at the ethics of biotechnology. So yeah, we focus on the social and economical implication of these processes. So the ethical positives of biotechnology are that, firstly, that they meet the growing needs of society, right? So they improve quality of life. For example, it meets the United Nations Sustainable Development Goals, and it can help in ending poverty and hunger. So now that we're referring here to the positives of genetically modified crops and genetically modified organisms like bananas and corn and BT cotton. So all of them really important and genetically modified, and so that can help us with ending poverty and hunger. It can improve health and well-being. So now, instead of, for example, with bt cotton, instead of spraying um, harmful insecticides to get rid of the pathogen, we have just genetically engineered it so that we don't have to do that anymore. um, yeah, so improving health and well-being, access to clean water and sanitation, clean energy, sustainable cities, harnesses biological tools to invent new creative solution. Genetic diversity is an interesting one, because by creating these new products you are creating genetic diversity, but we'll talk about the sort of the reverse of that very soon. Creating new arrangements of genes, new allele combinations, allows for organisms to adapt to their environment, ultimately accelerates the process of evolution. Open source information, and it leads to the creation of public databases to allow for biological knowledge to be shared. However, there are concerns regarding ownership. Who actually owns these technologies? Should we actually allow people or corporations to own the biological information? Economic benefits versus innovation. There are lots of economic benefits. Um, so lots of economic benefits, especially with monocultures. So the Cavendish banana, for example, is an example of a monoculture. So there's just that one species of bananas around the world, and that was because it was genetically engineered. Because the banana species before that, it sorry. So the banana species before that, it fell victim to the Panama disease, which is like a rust fungal disease, and affected banana populations. And that's why there was like, I can't remember, I think it was around 2000 and 2009, 10, 8, like around that category, where you would have seen banana prices like rocket, because of that. So after that, they created Cavendish bananas, which which was to sort of obviously, I think it took out the gene, or it sort of made it immune to that specific disease. But now there has been lots of concerns with that, because of the fact now, because it's now fallen victim to a new type of pathogen. Because there's a monoculture of bananas, there's no actual diversity in the species of bananas to fight that specific disease off. So yeah, so there's lots of economic benefits, but what is the price of innovation here? Intellectual property constraints can be prohibitive to process. And there is a case study on zinc finger technologies that you can read. There is also commercial implementation. Biotech monopolies is a big one. Companies with vast resources dominate the market, and they definitely drive on the processes of the products. Program dependency, consumer rights and choices. Does everyone want to eat genetically modified crops? What if they want organic crops? Regulation, governments, is there a balance between safety and innovation that they can strive towards? Biohacking and eugenics. So eugenics is an interesting new word that you'll learn today. And this is the idea that genetic technologies may be used to purposefully improve the genetic character of a population. And that is um, a very controversial practice. Past examples include World War Two. So World War Two is an example of that. During the World War Two, when there was the focus on like improving the genetic character of the population by like only um, by recruiting people and testing on people who looked a certain way. Um, so now, this table here is a really good source for that I recommend you guys have a look at. It's basically looking at fancy philosophy terms that you can use in your responses, and shows the mark that you sort of, especially with like these ethic questions, it's really good to have that vocabulary as well. Okay, so let's have a look now at module 7. I'll just grab a quick sip. Alright, looks like we're going well according to time. If you have any questions, please don't hesitate to ask. Okay, so key concepts in module 7. Causes. We won't quite look at causes today. We're going to focus on response, immunity, and prevention, treatment, and control. That's what we're going to focus on today. So we're going to talk about our first line of defense includes our skin, our chemical secretions, our chemical secretions, and also things like the toxins that we secrete, the antimicrobials we secrete in our skin. So I would recommend revising on that and remembering the 5 elements of the first line of defense. What we're going to focus on today is the second line of defense. And we'll start with the second line of defense and then we'll move into adaptive immunity. So the first and second line of defense is part of the innate immunity. So this is the immunity that is always there. It is not reactive. So it's not specific, rather, that's the better word. It's not specific to the pathogen, it's always there, and it's going to react in the same way. So the second line of defense includes, firstly, the lymph system. So the lymph system produces white blood cells that are responsible for the immune system. And with the immune system, it's not like one system somewhere in the body, kind of like your digestive system or your all of your other systems, your respiratory system, right? The immune system is a lot more mobile, and so what we see is that it's spread throughout the body, and the lymph nodes play a huge role in that. So we see that there's a lymph node, there's a network of tissues and organs that helps to get rid of the toxins and waste within our body, and that's the lymph nodes. So that's the first part. The second part is inflammation. So inflammation is the dilation of blood vessels and the infiltration of inflammatory cells. So what happens is histamine. So let's say you have hurt yourself, you've I'm just trying to think of something. Okay, you were at the park and you stepped on something sharp, and now you have gotten like a penetration, and it's bleeding, right? So what would happen immediately is that once you've done that, the pathogens would go in because you're at the park. Something's entered the body, some splint or something, it's entered the body, and it's obviously got pathogens on it because it's out in the environment. So now immediately your body is going to react to that. Your blood vessels will dilate, and your inflammatory cells, so these are called histamines, they are going to rush. So the histamines are going to cause them to rush to the site of infection, and then that's where your blood flow will be concentrated, and that's where you see swelling and redness, because all of these are coming to now deal with these harmful substances that have entered your body. So that's inflammation. Third step is phagocytosis. Now, phagocytosis is conducted by special white blood cells called macrophages and neutrophils. So what they do is they change their shape to engulf pathogens. So they basically eat up a pathogen and then destroy them using acids within their cells, like using like lysosomes. So these are lysosomes, they're going to come and destroy the pathogen within the cell. So that's phagocytosis. Now, do not confuse phagocytosis with apoptosis. So phagocytosis is when a macrophage or a neutrophil comes and it eats up the pathogen. Apoptosis is cell death to seal off the pathogen. So cells like macrophages and lymphocytes, they completely surround the pathogen, and after they surround the pathogen, they die. And so what happens is, once that happens, they form a cyst, which is then filled with pus, and it blocks pathogen movement. It blocks nutrient supply to that pathogen, so that it cannot grow, so that it cannot spread, and it dies. So that's apoptosis. So this is a regular cell, but then once we see it's been infected apoptosis is initiated, cell death. The cell is going to die so that it can take down the pathogen with it. Okay, so let's now have a look at what is the difference between the two. What is the difference between adaptive and innate immunity? So innate immunity is always there. It is essentially your body always has these mechanisms in place. As soon as something happens, these are going to jump at the chance to go and protect your body. Always there. Adaptive immunity, on the other hand, arises when a pathogen enters your body, and when it's not been dealt with the innate immunity, then the adaptive immunity sets in, but it takes time. The elements of the innate immunity include the first and second line of defense. The elements of the adaptive immunity include B cells and T cells, and we'll talk about that. So kind of like two families of cells that are going to be really, really important. Specificity. So innate immunity is non-specific. So it's going to react the same way to every single disease or to every single pathogen that enters your body. However, adaptive immunity is going to react differently to the pathogen, to every pathogen. So it's going to act specifically. So it's a very specific process. Sorry about that. Looks like my immunity's been doing stuff. Give me one sec. Alrighty. So, alrighty. Where are we? So specificity. Yep. So innate immunity, non-specific. Adaptive immunity, very specific. And so the thing with adaptive immunity is your body already has these T cells that are going to, like, it already has all of these basically, and we'll talk about this concept in just one second, but I want you to remember these antibodies to these specific pathogens in their place in the body. We just have to find them and react to the pathogen. So your body is like really, really cool in the fact that it already has all these defense mechanisms for things that it can encounter in the environment. We just have to find them, and that's what takes time. So that is why the response rate of the innate immune system, which sort of jumps into action straight away because it's doing the same thing every time it encounters something, is that it's very rapid, it's very fast. But with adaptive immunity, the response rate is very slow, or not very slow, but it's pretty slow, because on the first exposure, it is going to try and find the specific antibody for that pathogen. So a pathogen has an antigen on it, like a marker that tells the body that it's like a non-self cell, and so your body has antibodies that are going to react to that. And we already have all the antibodies, we just need to find it. That's why it's a slow response. But once we find it, we have something called the memory cells that are going to remember exactly what that antibody was to what specific antigen, and that will circulate in your body. And so the next time that you see that specific pathogen, your body is going to react really fast. So that is why innate immunity does not have any immunological memory, because it's reacting in the same way every time. Adaptive immunity indeed has immunological memory, because it remembers now how it dealt with that pathogen the first time around, and so the second time it happens, the response rate is really fast, it's really quick, and the concentration of the antibodies reaches a lot higher than the first time around as well. Okay, that's the key there, the key distinctions between innate and adaptive immunity. Let's move on then. So where are we? Okay, now we're going to look at T and B cells. So let's have a look. Probably let's start with T cells first. B cells are responsible for antibody-mediated immunity. So what happens is that B cells, there are two types of B cells. We've got where are we? My slides are working. Alright, we've got two types of B cells. We've got a plasma B cells, and then we've got a memory B cells. Plasma B cells, these are the ones that provide immediate protection. Alrighty, so they are the ones that provide immediate protection. They secrete lots of antibodies, and they have the same specificity as the parent cell. So this is after differentiation. So the B cell, it differentiates. So we've got the B cells, and then we've got the memory B cells. The memory B cells are the ones that have immunological memory. These guys are the ones that are going to remember to actually remember what that antigen was, so that and then they remain in circulation, so the next time that they encounter that pathogen, they can react immediately. T cells are responsible for cell mediated immunity. So there are four types of T cells. And I've just realized that we can't quite see the writing on there for some reason. Oh, there you go. You've got the plasma B cells and the memory B cells. Okay, that's funny. Give me 1 second. Let me see if I can highlight it. I don't know, for some reason the color looks different on my slides. So let me just fix that. Give me 1 sec. Where are we? Highlighter. And let's get... So we've got the oh no, that's even worse. Okay, so as I was saying, we've got the plasma B cells. These guys are the ones that are going to remember, that are going to immediately go ahead and secrete lots of antibodies. And memory B cells are the ones that are going to remember the pathogen for next time round. Then we've got four types of there we go, that's better. Not much better, but it's better. You guys can see it now. And then we've got four types of T cells. We've got cytotoxic killer T cells, we've got helper T cells, we've got suppressor T cells, and we've got memory T cells. So these are helper T cells. Helper T cells, as the name suggests, these are the nice guys of the group. They go and help other white blood cells that are involved in the process. And they secrete cytokines to help coordinate the immune response. Then we have the cytotoxic killer T cells. These are the T cells that destruct and release. They release cytokines to kill the pathogens by triggering apoptosis, by triggering cell death. They kill the cells that have been infected so that they can get rid of the pathogen. The suppressor T cells, think of them as like the best friend of the killer T cells. These are the ones that will then go ahead and control the immune response, and they'll control the immune response by suppressing the killer T cells that kind of want to go on a rampage. So that's what the suppressor T cells will do. And finally, we have the memory T cells. They're going to again remember what the antibodies were. They're going to remember what the antibodies were, and then as a result, they're going to remain in circulation to allow for quick response and a stronger response upon reinfection. And so that's why you'll see, and you'll get questions where I ask you, what is the difference between between primary and secondary exposure. So always remember, primary exposure, where can I draw? Okay, there's going to be, okay, just don't worry about this diagram for a second. Oh no, I've got on the slide after this, never mind. So I won't draw. I'll save you from my drawing. But essentially, primary exposure is going to be less stronger than the secondary exposure. And we'll have a look at what that means in 1 second. But let's have a look at what's actually happening now, what the whole process is. Let's put it all together. So, there is a pathogen. You have been infected with something. Let's say a flu virus. Your sister was sick a couple of days ago and you chose to hang out around her, and so you got sick as well. All right, not that it's a personal story, but it is. All right, so you got sick as well and you should not have hung around her, but you chose to because she was sick and you wanted to make her feel better. But nonetheless, so you got sick and now you're sick with a viral flu, and you're coughing and sneezing and all of that. So there's a pathogen in your body. It has an antigen on it. An antigen is like a marker that shows your immune system that, hey, and it's a way for your immune system to recognize that this is not a cell that belongs in my body. Like, it's come from outside. So that's like a strong red flag that we don't want that. So the pathogen, what will happen is, Our amazing heroic macrophages are going to go ahead and engulf the pathogen. They're going to eat the pathogen. Remember, these are white blood cells. It's sort of their role. They're going to go ahead and deal with this head-on. They're going to go ahead and eat the pathogen. And now once they eat the pathogen, macrophages have something called MHC class 2 presenters. Now your body has MHC complexes. Class 1 is present on all body cells, and it's basically a way for your body to show that, like for example, the proteins that it displays on its surface to show your immune cells that via self cells. Macrophages have MHC class 2 presenters, and so once the macrophage has eaten a pathogen, it is going to display the antigens on its surface to show the body that, hey, we've got something entering our body, and this is what it looks like. This is the antigen. This is the marker for it. And so once that happens, we're going to have these cells called interleukins that go ahead and they're going to call helper B and T cells to the site of function. So, and once they've done that, now our helper B and T cells are down and ready to go. So what that means is this: once they're ready, and B cells are going to be part of the antibody mediated immunity, and helper T cells are the cell mediated immunity. So what happens is once the B and the T cells are there, essentially the helper T cells are going to come and they are going to try out different antibodies until they find the right antibody for the antigen. Same thing with the B cells. Once they find the right antibodies, they're going to go ahead. B cells are going to differentiate into plasma B cells and memory B cells. Plasma B cells are going to secrete lots and lots of antibodies to immobilize the pathogen. So they're going to go ahead and immobilize the pathogen like that. And once they immobilize the pathogen and they, like, the antibodies go ahead and stick to the antigen, the killer T cells come and they kill these off because they now see that essentially these are the things that are, you know, that are causing that, like that's basically causing the disease. So these are pathogens, these are foreign invaders, we don't want that. So they come ahead and so they're going to secrete perforin, which is like a chemical to destroy off the pathogen. At the same time, the memory B cells are going to remember what antibody worked for this specific type of antigen and they're going to store it in the lymph nodes. Helper T cells are sort of doing the same thing. They're going to come ahead. They've found the antibody. They're now going to send cytokines. So it's called cytokine signaling. They're going to signal cytokines to go ahead, and then they're going to call killer T cells to the spot. They're going to call suppressor T cells because wherever killer T cells go, they need suppressor T cells. Otherwise, killer T cells can go on a little rampage. And then you've got memory T cells, which are also going to store the antibody in the lymph nodes. So here, killer T cells, and then we've got suppressor T cells, and what happens is they suppresses killer T cells once the immune response is finished, and we've discussed that. And after a while we've dealt with the pathogen, and most importantly our memory cells remember the antibody that was needed for it, and that's so important. So that's done and we have dealt with the pathogen. So that was immunity. If you have any questions, please ask. Again, it was a little review into it. If you have any specific questions, like I said, don't be afraid to ask, and definitely make sure that you've got detailed notes of these processes because it's really important. Alrighty, let's have a look at this last point about prevention and control. But before we do that, let's have a look at this diagram that I've been sort of trying to explain with my hands the whole time. So, this is basically how vaccines work now. What vaccines do are essentially within vaccines we add in attenuated, dead, or synthetic viral particles of a specific pathogen into the vaccine, right? And then we do that so that we can have a primary immune response. It's not the actual pathogen, so you're not like sick as such, but that's why you experience symptoms that are specific to the disease. So like coughing and so on, or be having a fever or being sick or being tired, is because we are trying to induce that same response. So When you get that first vaccination or infection, after a couple of days, your antibody count goes up because your body has found a way to deal with it. So what that means is that the next time round when you actually get the pathogen, when you're actually infected, your antibody count will shoot up. The response will be faster, right? So as soon as we get you can see here that it took us what, when you initially get the vaccine, like up to like 25 days, I would say, right there. Or yeah, the peak, yeah, 25 days before you actually peaked and you got that high antibody concentration response. In comparison to the second time round, where it took just about there, that's the sort of the high there, about like 20 days as well. So less time, but a lot higher antibody response. Very, very high. Immediately, just a couple of days afterwards, the response was so much higher than it had ever been the first time around. So that's how vaccines work. So that so that when you actually get the the pathogen, when you actually come in contact with it, your secondary antibody response can be really fast, and so you don't get as sick. So when it comes to um, prevention, treatment, and control, some really important things include hygiene practices. So washing your hands, coughing when, like, putting an elbow when you're coughing, or sneezing and not, like, just sneezing normally. That was high, and you'd remember this from COVID-19. So hygiene practices. Then quarantine was quarantining, not going outside. If you're sick, stay home. And even generally, we had everyone quarantine, so that there's no exchange of pathogens there. uh, Vaccination was very important, and it was part of the public health program. Pesticides comes in, like pesticides, for example, obviously weren't used for COVID because, it's a viral, so not used for COVID. However, pesticides, for example, really, they were a key part of the response, for example, for malaria. Spraying pesticides, huge. And genetic engineering. So genetic engineering is something that is, still sort of, in progress as a process. And so this is where we are engineering resistance to pathogens, so to plants, animals, vectors, humans. And there's been research into, like, engineering Anopheles mosquitoes that spread malaria, and engineering so that they don't have. I cannot remember. I think it's like they're like red eyes or something. I remember the image that was in that article. It was like a close up of a mosquito with red eyes. It was a bit scary. No, but it was basically how they were going to engineer these mosquitoes so that they cannot carry the protozoa anymore. Um, and so basically that's to modify those genes so that they can stop that, the spread of the disease, because vectors, that's the way it's spread, and it's huge. It's a huge issue worldwide, as well. Okay, we're going well according to time, so let's have a look now at a last module. Module 8. So we're looking at non-infectious diseases, and we're going to focus on homeostasis, epidemiology, and tech and disorders. So homeostasis. There are two homeostasis mechanisms that you have to know according to the syllabus, and that's your temperature, and glucose. So I won't do the temperature one because I think that one's pretty obvious. You know what happens when your body temperature goes high? You start sweating, your blood vessels dilate. Same thing happens. Oh well, the opposite happens when your body temperature decreases. You start to shiver. Your blood vessels constrict so that you don't lose heat by blood, and so on. So essentially, that's the temperature homeostasis. So hopefully you are familiar with that. The one that I'm going to focus on, because it has a lot more going on, is the glucose homeostasis. So there are two parts to it, and we're going to let's start off with homeostasis. So normal blood glucose level, here it says it's 90 milligrams per 100 mils. You can also remember it as like 45 millimolar. Uh, that's perfectly fine. Like 5 milli, 4 to 5 millimolar. Anything below 4 is when you have to get like when yeah, it's not good. So what happens is, let's say your blood glucose level rises, right? Your blood glucose level rises. You've just had a meal. You had pizza and soft drink for lunch. And I don't want to think about pizza because I've got a sore throat and I cannot eat. But let's say you've had pizza. So you've had a pizza, you've had a soft drink, and it's very high in carbs, cheese, fat, so on. Soft drink very high in sugar, and your blood glucose level has shot up to 8 to 10 millimolar. That's what usually happens after a meal. It shoots up. So once that has happened, what we see is that your insulin secreting cells of the pancreas, your islet cells in the pancreas, beta cells, what they do is that they start to release insulin. And insulin is a hormone that is responsible for actually bringing your blood glucose level back down. So what insulin does is that once it's been released, it goes ahead and it signals mainly the liver, but also the muscle sometimes. So mainly the first place it will go to is the liver, and it would say take up some glucose from the body. We've just got a lot of glucose. It's not good. It can really affect. And the reason high glucose is not good is because it can affect cell function. It's just not good overall. Over a long period of time, it can affect like other functions of the body as well, and it can have huge impacts on, for example, the heart. And function of the heart, and also like your respiratory system. So that's why high blood glucose level is something that we don't want to have, and that's what doctors always suggest against. So insulin has left the pancreas. It's been secreted by the pancreas. It goes to the liver. It says, hey, take up some glucose and store it as glycogen. Glycogen is basically like a molecule that has like 10 to 12 glucose attached to it. So it's kind of like, think of it like as a, yeah, it's like basically like, oh, what am I pressing? Um, yeah, like it's got gliadin in the middle, and then it's just got glucose attached to it in a tree formation. So each part like branch is a glucose molecule. So that's what basically um glycogen is. It's basically like a huge store of glucose. So every molecule has like 10 to 12 molecules of glucose attached to it. So what happens is essentially that once your insulin goes, it tells the liver to take up glucose and store it as glycogen, and then blood glucose levels go down to that 45 millimole over a period of time. It can also ask the body cells to take up glucose as well. And so we come back to our key sort of 4 to 5 millimolar levels of glucose. However, then let's say that you haven't had a meal in like 3 hours. Alright, you haven't had a meal, you haven't had anything to eat, so immediately your blood glucose levels down. So then, this time around, your pancreas are going to secrete, the alpha cells in your pancreas are going to secrete glucagon. And glucagon is essentially going to go to the liver and say, hey, all the glycogen you had, you've got to break it down into glucose and send it throughout the body. And once it does that, because your body needs glucose, your cells need glucose, your brain needs glucose a lot, your brain uses a lot of glucose. So it's saying, go ahead, go circulate glucose to the body, and then the glucose goes ahead, and as a result, your blood glucose levels rise to that set point again. So this is really important in understanding how we come back to the equilibrium level. So like this is about knowing that regardless of the external conditions, your internal environment has to stay in a certain state, and that's what the glucose homeostasis is about. So the key organs here are the pancreas, which detect glucose levels and they secrete hormones. Insulin is secreted when glucose levels are low, and it's secreted by the beta cells. Glucagon is secreted by the alpha cells in the pancreas. Liver stores glucose as glycogen, and then it also releases that glycogen, and other tissues and cells, they use up glucose. And so that's that. And then finally we've got hormones. So glucagon is a low blood glucose hormone. It's saying that, hey, you've got to release some glucose now. Insulin is high blood glucose. Something that and this is something that you also sort of look at when we look at type 1 diabetes in this module. You don't quite study it as such, but you look at, you know, how its high prevalence and so on. All right, so you look at its high prevalence, and the reason why type 2 diabetes, because type 1 diabetes is an autoimmune disease, and so that's because in type 1 diabetes, your body's your immune cells start to attack your beta cells, and so they start to attack the insulin producing cells, the insulin secreting cells rather, and as a result you can't produce that insulin, so you can't control your blood glucose levels. What happens with type 2 diabetes is that over time, if you keep having sort of unhealthy meals and keep having unhealthy meals, and your body sort of, especially as you get older and your body has to release more and more insulin to deal with that, to bring your like glucose level back to normal. Over time, your body can become resistant to insulin, and it goes from slowly insulin resistance to then like not reacting at all to insulin, and then that's type 2 diabetes. And that's why they say, exercise and healthy eating is like key to controlling type 2 diabetes, especially when you're sort of showing signs of it at the beginning. So that's the glucose homeostasis. Okay, let's move on then. I cannot find my cursor. Give me 1 second. Where is it? There we go. Okay, so epidemiology. Let's have a look at epidemiology. So let's consider what makes a good or a valid epidemiological study. So your goal is to always design a study where you're going to generate a lot of data to conclusively test your aim. So you can be given like a study in a question, and they can ask you like, is it evaluate the validity of the study or something. So always remember that a good study should have a large cohort or a sample size, right? It should have a broad cohort as well. So you can't have the same sort of type of people if you are testing. Let's say if you're testing heart, and this is a very random study and very broad, but let's say you're testing heart conditions and lack of exercise in individuals, like the relationship between heart conditions and exercise, and you're just testing young people, you're not going to actually get much, because it's like young people are not that, they obviously exercise a bit more, and also heart conditions generally, like older people are generally susceptible to it. So you want a broad cohort, in that older people category, which includes broad, that people coming from a diverse range of environment, ethnicity, exposure to possible causes as well. You also want to conduct relevant background medical checks for participants and create a comprehensive questionnaire, and peer review prior to publication. So good studies are peer reviewed. So essentially collect enough information so you can see effect of different variables. And this checklist is really good. So commit it to memory, you'll need it, and use this to evaluate different studies as well. Okay, analyzing patterns. So here we'll have a look at when it comes to analyzing patterns in epidemiological study, we consider things like, what is the difference in incidence between different countries, what is the difference between different incidence between genders, age groups, lifestyles, occupations, and that is used to draw conclusions. Okay, last part of module 8, technology and disorders. So alrighty, let's have a look at this. So, when it comes to eye disorders, we are looking at, firstly, this is what a normal eye looks like. I won't go through the structure of an eye, but I'm just going to show you, that is the structure of an eye. So we've got a normal eye here. This is the focal plane. So the focal plane is at the back of the retina there, where the light hits the back, just where your cones and where your cones and rods are. That and they are the ones that are responsible for changing the image into sensory signals, which is then sent to your brain, right? So that's the focal plane in a normal eye. In a myopic eye, so myopia is nearsightedness. It's when you can see things close to you perfectly well, but things far away are blurry. So this is where this happens when the cornea is too curved. So the cornea here, you can see here, it's like longer, less curved. Here it's too curved, and because the cornea is too curved, what happens is that the light doesn't go all the way back. So the cornea here is too curved, so light does not go all the way back. It should be hitting, this is where the focal plane should be, the light should be hitting all the way back here, but because it's only hitting the middle of the eye here, we can't see objects that are far away. Hyperopia is when the focal plane is actually behind the retina, and because now it's behind the retina, we cannot see. This is called farsightedness. We can see things that are far away very clearly. We can see them very clearly. However, things that are closer to us, we can't see them very clearly because of this idea here of the focal plane being far too back. And so that is the difference between myopia and hyperopia. So causes are in myopia, the cornea is too curved, so the image doesn't go all the way back, it hits most of the front. Hyperopia is the cornea is too flat, so the eye it's too flat, it goes all the way back, and so it could be if the eyeball is too long or if the eyeball is too short. So to correct that, generally we wear glasses. So myopia is when people wear, to help with myopia, nearsightedness, people wear concave glasses. So that's like to help the image go further back, so to help the focal plane move further back. So it's like flatter glasses. Hyperopia, because the cornea are too flat, with our glasses they're convex, so that the image does not travel behind the focal plane, so it hits like behind the retina. So that's that. Now, if you want to have laser eye surgery done because of that, laser eye surgery would involve rectifying the cornea shape, so flattening the cornea because it's too curved, or increasing the curve of the cornea because it's too flat. And that's myopia and hyperopia. Now, the E. So, with the E is divided into three parts. We've got the outer E, and the outer E includes the pinna, the ear canal. So we've got the pinna, the ear canal, and this is how sound waves enter the ear canal. So that's the outer E. The middle E is where we've got the eardrums. We've got the eardrums here, and we've got the ossicles. So this bit here is that's the ossicles, and I'm gonna rub that off so you can actually see what they look like. There you go. That's the ossicles right there. And so that's the middle ear. And now what happens in the middle ear is that when the so when the sound waves come through, they vibrate against the eardrum, and when they vibrate against the eardrum, it beats against the ossicle, and it moves the ossicle bone, and so the ossicle bone will it's kind of like open close in a way, but it will kind of open up and close and beat against the cochlea here. It will beat against the cochlea over here. So you've got the eardrum, you've got the ossicles, you've got the cochlea there, the cochlea here, and so the ossicle will beat against the cochlea here. And then the inner ear includes the semicircular canals, it includes the cochlea, and it includes the auditory nerve. So what happens is, once the ossicles beat against the cochlea have they've got a fluid in there, and they also have little hair that line the cochlea, and it's the little hair that are actually responsible for changing the frequency of for understanding the frequency of different sounds, for not understanding, what's the word, for converting the frequency of different sounds into like electrical waves. So the cochlea actually does the conversion of the sound waves into electrical impulses, and then those electrical impulses are sent to the brain via the auditory nerve here. So you can see how the cochlea is connected to the auditory nerve there. So that's that. So that's how hearing works. So to rectify disorders of the ear, we've got 3 different technologies. Now you need to remember which technology would suit in which case. Conductive hearing loss. For conductive hearing loss, essentially, we've got conductive hearing loss is when it's affecting the outer and the middle ear. So hearing aids work for conductive hearing loss because what they do is that they magnify the sound vibrations that are entering the ear. They increase hearing sensitivity, and they can be worn inside the ear or behind the ear. So they basically magnify the sound vibrations that they go all the way through. So here they are. Bone conduction implants, they can be used for conductive hearing loss, so outer or middle ear, or also for mixed hearing loss, which is mixed. So what that means is, so mixed hearing loss, sorry, is both outer and inner. So what that means is the sound waves are detected by the processor. There's a processor that detects the sound waves, and it actually converts the sound waves digitally into vibrations, and then it sends the vibrations to the implanted section of the device, and that stimulates vibration of the ear bone. So that's what the bone conduction implants doing. Finally, cochlear implants. Now, cochlear implants, this is for sensorineural hearing loss. So this is for the inner ear damage, and now this is basically replacing the function of the cochlea. So what happens is that you've got sound which is received by an external transmitter, and it's turned into digital code, and it is converted to electrical impulses. So you've got the microphone, it picks up the voice or the sound, right? And there's also a speech processor. So the speech processor actually processes that, and it changes and it turns the transmitter turns the speech processor turns the sound into a code, and then through that it transfers so it turns it into a code, it turns it into electrical impulses, and then it transfers that specifically, as you can see here, the implant on the inside of the head, on the inside of the ear here, it goes straight through to the auditory nerve, to the cochlea. That is what's happening here. So this is where, because the inner ear is damaged, the cochlea is not working, so we have to somehow get the sound vibrations, like we change them into electrical impulses and get those electrical impulses from the implant to the auditory nerve. And so you can see how the implant inside the head here is connected to the auditory nerve, or to the cochlea, and then here to the auditory nerve, and going straight through. Okay, so finally the kidneys. So we now remember the structure of the kidneys. So these are the medulla, you've got the cortex, you've got the renal pelvis here. This is the renal artery, and this is the renal vein. And then you've got the ureter, which connects down to the urethra and the bladder. So there are 2 disorders with kidneys, and this is showing you what a nephron looks like. So there are nephrons here within the cortex and the medulla, and this is what a nephron looks like. This is where the filtration happens. So we've got the glomerulus right here within the Bowman's capsule. You've got the glomerulus, and this is how the blood the filtrate comes through, and so it comes in, goes through the proximal tube, down the loop of Henle's. There's the ascending and descending and ascending loop of Henle, up through the distal tube, and in the collecting duct. So the 2 disorders of the kidneys are, first is a polycystic kidney disorder, which is a genetic disorder which causes growth of fluid-filled cysts in the kidney. And then we've got chronic kidney disease, which is the gradual loss of kidney function over time. So the technology that we use for kidney that can be used to like, I guess, manage kidney disease is hemodialysis and peritoneal dialysis. But the only way to like manage it long term is to get a kidney transplant. So dialysis is basically removing the metabolic waste, the salt and the extra water from the body, which the kidneys do internally. It's doing that externally by allowing dissolved solute to pass through a semi-permeable membrane. So what happens with hemodialysis is we take the blood, it's pumped through this machine here, and basically as it goes through osmotic pressure, we're taking out the waste here. This is the used dialysis fluid, and we're set and fresh fresh dialysis fluid is being sent into the machine, and so we're taking out the waste, and through that we are clearing the blood of the metabolic waste and sending it back through into the body. Peritoneal dialysis does this inside the body. So hemodialysis is essentially hemodialysis, like to have hemodialysis done, you have to go to a hospital and use the machines there, and it's definitely very time and cost and energy expensive. Peritoneal dialysis works in the same way, but it's actually working within the abdominal cavity, in the lining of the stomach, so the peritoneum. And so we've got a dialysis solution. We use a catheter to have that process of filtration done within the body. But essentially the key here is that you can talk about these processes. They are effective, but they're obviously not as effective as what the actual kidney does. And they're definitely very time expensive, very costly, and very energy expensive as well. So yeah, so you want to think of those points when you are writing responses and you're commenting on the effectiveness of these machines and these processes. But remember, yeah, the with kidney disease, the long-term management is a kidney transplant. So you do this until you can get a kidney transplant. Alrighty, so look at us go. Alrighty, so we have gone through the syllabus. We've gone through the main parts of the syllabus. Now, if you have any questions, please ask. The live chat always closes a couple of minutes after the lecture finishes, so please ask any questions that you have. But let's have a look at how you can now prepare for the final exam. So my key tips would be, firstly, practice papers. Now is the time to extend yourself. Redo the questions that you struggle with. Don't just do it and leave it in there. Mark these questions. Really, really important. Mark these questions. Then refine your notes. So this is where focus on key terms. Like, make sure you have all the necessary examples, and use these in your practice. But the thing is, I would recommend, if you are now if you're coming towards the last, this sort of tail end of the the exam, like prep period, I would recommend to focus more so on practice papers than refining your notes. Like, make sure you have all the relevant notes, but like, don't spend time sitting down and trying to make them pretty and like really succinct and whatnot. Do practice papers instead. So if you're choosing now between refining notes and practice papers are the way to go. But make sure you've got, like, really important is that you've got all the necessary examples. Then write or plan long response questions. So that is basically really important. You want to write clear example responses to all long questions. It's a really good exercise and it makes you feel really confident. And you can access oh, goodness. Okay. And you can access lots of questions if you search up NESA additional sample examination materials. So that's basically when the new syllabus change came, NESA posted this like 80-page document that has all these practice questions that are like labeled according to different bands and modules. So access that. It is really, really good. Now, practice analyzing questions and planning responses. Focus on implementing the key terms and context. Focus on structure. Focus on structure. Focus on like answering like the verb. So like write as much as needed. Really, really important. Use examples. Use use key terms. What else was I going to say? And also make sure when you do the additional sample question material, you also practice there's a lot of like graph questions as well. So remember, Nessa is big on skills. They're gonna give you graphs. They're gonna ask you to interpret data. So make sure that you feel comfortable doing that. Okay, tips. Okay, tips. So there's a video for you to have a look at, to have a which discusses exam strategy. So just to help you guys choose an exam strategy for you. I'm not sure of how you'll approach the exam. Yeah, I personally, I just did it back to front, but like the thing I always do is that if you don't get something, leave it, come back to it. Really important you do that. Even if you feel like you have no idea what you're doing. Okay. Even if you have no idea what you are doing, leave it, come back to it. You'll know somehow. That's just how the brain works. It's really cool. So just leave it, come back to it. Don't get caught up in questions you can't ask. You can't answer. Plan all your responses, even shorter ones, and especially the long ones. Plan them out. Identify the syllabus area. Use key terminology. Identify harder questions. So definitely make sure you spend time, like extra time on this harder, 7, 6, 8 mark questions. There was a 9 marker in the 2021 paper, sorry. Scientific drawings, clear lines, do not shade in between, no shading, and no shading, but label. That's really important. And then finally, graphs, Punnett squares, pedigrees. Always keep it simple, keep it neat, and give them a key as well. Always include a key, especially for Punnett squares and pedigrees, and also for graphs if you're using different symbols. And once you finish the answer, go back, reread the question. Did you actually answer it? So a tip that we like to use here, like to do practice questions at TuteSmart, for example, in my class, is like 2 minutes per mark kind of thing, and that's just calculated like on 180 divided by 100, which gives you 180 marks divided by 100, so it gives you about 1.8 minutes, and so we just sort of go like 2 minutes essentially per mark. So yeah, look, that is something that you can use if you like, or you can find your own little trick that works for you. But the 2 minute per mark thing just gives you about 20-second per mark to the like add into your account, and then you can come back and read the paper again, or read a question again, or spend extra time on a question. But see what works for you. Go in with a plan. Go in how you're going to attack the question. Don't just sort of be like, okay, I'll see how it works in the day. And this is where doing past papers beforehand really helps, because you've done them beforehand. You know exactly what works for you, and that's exactly how it should be. You should know what works for you. So that's that. For your checklist, make sure you have an intro sentence plus a judgment sentence. Define the key terms. Name examples. Make sure you answer the question. Really important. Have a concluding sentence. So this is generally for all of your responses, but for long answer responses, read and carefully underline all key terms. Make sure you identify the syllabus area as well, and then plan a structure. Do not remember, you want to spend, even if it takes you 1, 2, 3 minutes to plan a structure before you start writing, you wouldn't do that instead of jumping straight into writing. So plan a structure. Make sure you have a strong introduction and conclusion. Use diagrams, tables, flowcharts if you have time. And then don't be afraid of subheadings as well. Like, you can use subheadings if it helps. And finally, read the question after every paragraph. Structure is so important in bio, so important. So make sure that you actually plan it out, kind of plan it out like a little essay, so that you're covering every single thing. I'm just thinking if there's anything else I've missed. No, look, hopefully I've answered any questions, but now in these next couple of days, if you're struggling between choosing to either rewrite and make your notes more succinct to doing practice questions, do practice questions. Even if on days where it doesn't feel, where you don't feel like, and you will have those days, everyone has those days, even if it's close to the HSE, your body doesn't know that it's stressed, it's going to need those days. So even if you cannot sit there for 3 hours and do the whole paper, do one question, do two questions, do half an hour worth of questions, and then leave it. But do something, do a little every day. That can be really helpful. What else? Yeah, if you're struggling between rewriting your notes and practice questions, always practice questions. But if there are parts of the syllabus that you don't have notes on it, that you're not like familiar with, definitely do those first. One key thing that really worked for me with STEM subjects was like using a traffic light system. So that's basically when you look at your syllabus and you highlight things red, green, or yellow. Red is where you have no idea what you're going to write for it. If you get a question on that topic, more than likely you would probably not be getting any marks. Yellow is when you've got some idea, you'll be able to write some things but not everything, so you may scrape a couple of marks. Green is like, I know this topic really well, I wish the whole question was about this, the whole paper was about this specific topic. So go through the syllabus and highlight each point, red, green, or yellow. Now, don't underestimate yourself and do everything red. And I say that from experience because I did that, and then I realized, oh, that's not very good. And then I realized I was being tough on myself. So do not be tough on yourself. Think through it. Like, there will definitely be green areas, but there definitely be red areas, and that's okay. So do that and then prioritize your learning according to that. Do practice papers. But yeah, look, that's it from me today. Thank you guys for tuning in. Good luck. You got this. You're going to smash it out. Look after yourselves. Enjoy this. It's going to be over soon and you're going to have a lovely, amazing time after that. So take care. You guys are going to smash it. Good luck. And I will see you guys next time around.